So today is the second part of the same topic, that is the enzymes. Now coming to the next part of the chapter, that is the biomolecules, that is factors affecting enzyme activity. So what will be the factors affecting enzyme activity will be whenever there is a change in the enzymes, and enzymes we said are what? Proteins. So any change in the protein structure will affect the enzyme activity. So that is what we have said, ki activity of an enzyme can be affected by a change in condition that is going to alter the tertiary structure of the protein. And what are the factors? Let us see. First is the temperature. So temperature है कि अगर हम temperature increase करेंगे तो rate of enzyme reaction will increase. लेकिन there is a optimum temperature at which the enzyme is going to show highest activity. So the temperature at which an enzyme shows the highest activity, what do we call that as? Optimum temperature. And the optimum temperature क्या range है? Between 25 degrees to 35 degrees Celsius. But what I said, activity will decline. अगर increase करेंगे temperature को तो what will happen to the enzyme? It is going to get denatured. So there will be no activity. And what happens in denaturation? कि जो weak bonds है holding the protein, that is the enzyme, in the three-dimensional state, so that will break and it is going to unfold. The protein is going to unfold and it is going to, as a result, lose its activity. So that is what we say as denaturation. अगर temperature below the optimum होगा तो that is also going to reduce enzyme activity. क्योंकि अगर फ्रिज में हम कुछ food stuffs रखते हैं, why do we do it? Why does it remain fresh? Because the activity of the enzyme gets stopped and it does not spoil the food. But if we take it out and get it to the room temperature, the enzyme's activity will get resumed. So that is what we say, कि low temperature will preserve the enzyme in a temporarily inactive state. But if we increase the temperature, the activity of the enzyme will again start. लेकिन high temperature से क्या होगा? That is going to destroy the enzyme activity because it is going to break the hydrogen bonds holding the protein in the three-dimensional state and the protein will not function. It is going to lose its activity. So what we say, that the proteins will become denatured by heat. So this is a graph that we show one side is the enzyme activity and the other side is the temperature. So with the increase in the temperature, enzyme activity will increase and its maximum where? At the optimum temperature. So what is the optimum temperature? The temperature at which the enzyme activity is at its best. But if we either decrease or increase the temperature, so the activity is going to decline. Same for the pH, effect of pH. So enzymes are best functioning at which? Optimum pH. So here also increase in the pH or decrease in the pH will reduce the enzyme activity by altering the ionic bonds. और most of our जो intracellular enzymes है जो cells में function करते हैं वो near neutral pH में act करते हैं. But you have exceptions, जैसे कि digestive enzymes you have read, some are active in the acidic medium like in the stomach, like pepsin, but most of the enzymes in the small intestine, they are active in the alkaline region. So examples here, salivary amylase, you already read, it functions best at the pH 6.8, trypsin 8 पे और pepsin at pH 2. यहाँ पे भी same है कि enzyme activity will increase and will be at the most at its optimum pH, but increase or decrease in the pH is going to decrease the activity of the enzyme. Now coming to the concentration of substrate. So substrate में कि अगर हम substrate की concentration increase करेंगे तो velocity जो enzyme reaction की है वो भी increase होगा. लेकिन एक स्टेज आएगा which we call as the maximum velocity, तब reaction velocity भी constant रहेगी even if we increase the concentration of the substrate. As we can see from this diagram, ये यहाँ से velocity of the reaction है which is as capital V और यहाँ से substrate concentration है. तो we have increased the substrate, so with the increase in the substrate concentration the reaction has increased, मगर अब फिर flat हो जाता है, it reaches a plateau. And यहाँ पे इस stage पे even if we increase the substrate concentration, the velocity, the maximum velocity of the reaction will remain the same. It's not going to increase. Why? Because all enzymes, there are no free enzyme molecules to bind to the additional substrate molecules. So that is what we are saying here, that the reaction reaches a maximum velocity which we say as V max, after which the reaction velocity remains constant even if the concentration of the substrate is increased. अगर हमने increase भी किया फिर it remains constant. Why? This is because all enzyme molecules binds to the substrate and there are no free enzyme molecules to bind to the additional substrate molecules. Also we have shown is Km here. What it is Michaelis-Menten constant. And what is, how do we define that? It is of the Km value of an enzyme. What is that? Is the substrate concentration at which the enzyme catalyzed reaction reaches half of its maximum velocity. So this is half of the maximum velocity. This is the maximum velocity here. This is the half of the maximum velocity which we depict as half V max. So obviously अगर Km वैल्यू लो होगा, सपोज Km वैल्यू इतना होगा, लो होगा, कम होगा, so what does it show? कि जल्दी से बाइंड हुआ है, that means the enzyme has, that the substrate has got attached to the enzyme very quickly and they have reached the half velocity, half of its maximum velocity very fast. अगर very fast होगा तो affinity between the substrate and the enzymes is much more. Higher affinity है, अगर higher affinity है binder quicker होगा between the substrate and the enzyme. If binding is quicker, they are going to reach the half maximum velocity faster. So Km low, Km value, what does it indicate? Higher affinity between the enzyme and the substrate. लेकिन अगर high Km value होगा तो वो what does will it indicate? Weak affinity between the substrate and the enzyme. क्योंकि अगर high values होगा Kmk, that means half the maximum velocity पहुंचने में उसको टाइम लग रहा है. अगर टाइम लग रहा है तो affinity between the substrate and the enzyme will be weak. So that is what we are showing in the next slide. That is Michaelis-Menten constant Km. What did we say Km is? Km of an enzyme is the substrate concentration at which the enzyme catalyzed reaction reaches half its maximum velocity, that is half V max. And we also said कि एक low Km अगर होगा, that indicates high affinity. अगर high affinity है तो binding between the substrate and the enzyme will be quicker. So it will reach to its Km value much more faster, half of its maximum velocity much more faster, and it will have low Km. So that shows high affinity between the enzyme and the substrate. अगर high Km value होगा तो वो क्या reflect करेगा? Weak affinity between the enzyme and the substrate. And we have also seen in the previous slide कि जो graphical representation था of the reaction velocity versus substrate concentration, what was it? A rectangular hyperbola. Next will be the activators. So what we have seen, some enzymes require certain inorganic ions like magnesium, जैसे DNA polymerase है, manganese, zinc है, जैसे कार्बोक्सीपेप्टाइड है इसके लिए देखा है, कैल्शियम है, क्लोराइड है for their optimum activity. Next part of the enzyme is inhibition. Inhibition means reduction or stoppage of enzyme activity. So that was what we are going to do in this this topic, that is inhibition of enzyme action. So reduction or stoppage of enzyme activity due to a presence of certain chemicals, जिसको हम क्या बोलेंगे, inhibitors, is called as enzyme inhibition. अब enzyme inhibition 3 categories में है. पहले है competitive inhibition, next is non-competitive inhibition, third is allosteric inhibition. So first coming competitive inhibition meaning competition होगा किसके बीच में, इनहिबिटर और सबस्ट्रेट के बीच में. क्यों कंपटीशन होगा, क्योंकि जो इनहिबिटर का जो मॉलिक्यूलर स्ट्रक्चर है वो किसके साथ रिजेंबल करेगा, सबस्ट्रेट के साथ. चूंकि इनहिबिटर और सबस्ट्रेट का सेम मॉलिक्यूलर स्ट्रक्चर है तो वो दोनों किसके साथ बाइंड कर सकते हैं, टू द एक्टिव साइट ऑफ द एंजाइम. तो अगर इनहिबिटर ने बाइंड किया टू द एक्टिव साइट ऑफ द एंजाइम तो उसके रिजल्ट से सबस्ट्रेट कैन नॉट बाइंड टू द एंजाइम. तो अगर सबस्ट्रेट ने नहीं बाइंड किया एंजाइम्स के साथ तो कोई रिएक्शन नहीं होगा. सो एंजाइम एक्टिविटी इज गोइंग टू डिक्लाइन. सो दैट वी आर शोइंग हियर. अगर ये एंजाइम है नॉर्मली इट बाइंड्स टू द सबस्ट्रेट, बट इफ देयर इज एन इनहिबिटर जिसका मॉलिक्यूलर स्ट्रक्चर मैच करेगा किसके साथ, सबस्ट्रेट के साथ, तो इंस्टेड ऑफ द सबस्ट्रेट बाइंडिंग टू द एंजाइम व्हाट इट इज बाइंडिंग टू द इनहिबिटर. So when the inhibitor is going to bind to the enzyme, so it's blocking the active site. So as a result, substrate cannot bind to the enzyme. So if the substrate cannot bind to the enzyme, there will be no reaction. So that's what they have shown here. Substrate cannot bind to the enzyme because of the presence of this competitive inhibitor here. So this diagram is about non-competitive inhibitor. It should be till here only. So that's what we are saying. The inhibitor competes with the substrate. Initially what we said, that the inhibitor I, that is we have denoted as I, closely resembles the substrate in its molecular structure, and because of that the inhibitor competes with the substrate and binds to the active site of the enzyme. As a result, the substrate cannot bind to the enzyme and the enzyme does not undergo any catalysis. So enzyme activity is going to decline. But competitive inhibition generally reversible होता है, and अगर हम substrate की concentration increase करेंगे. So in competitive inhibition जो है Km value increases because affinity between the substrate and the enzyme थोड़ा कम होगा, affinity कम होगा तो Km value increase होगा. But Vmax will remain the same because जब काफ़ी सारा substrate का concentration होगा, the reaction still will be completed, because we said when this can be overcome, competitive inhibition can be overcome when we increase the concentration of the substrate. One example of competitive inhibition is the inhibition of the enzyme succinic dehydrogenase. And this inhibition, this succinic dehydrogenase is an enzyme that you are going to read in the citric acid cycle and the electron transport chain. So I think that chapter is going on presently, and this enzyme, what it does, it is removes hydrogen from the succinic acid and formation of fumaric acid or fumarate. अब इसका inhibitor कौन है? दो हो सकते हैं, या तो malonic acid और malonate होगा, या oxalic acid और oxalic acetate होगा. Now this I shown, there is no need for you to draw these structures, but I just want to emphasize कि देखो तीनों को, this is the normal succinic dehydrogenase, उसका क्या formula है कि it has a carboxylic group at the two ends, CH2 and CH2. ये succinate है, that is the normal. अब जो oxalic acetic acid है, जो हमने oxal acetate बोला as an inhibitor है, इसका भी structural formula same है. So it has two carboxylic group at two ends, only it has a carbonyl group here and CH2 here. Malonate का भी ले लो, देखो carboxylic group है, COH group at two ends, एंड एक CH2, तीनों का जो मॉलिक्यूलर स्ट्रक्चर है मैच करता है. सो ये नॉर्मल, ये है सबस्ट्रेट है. दीज टू आर द इनहिबिटर्स, कॉम्पिटिटिव इनहिबिटर्स, क्योंकि इनका केमिस्ट्री मैच करता है विद द सबस्ट्रेट. सो दीज कैन ऑल्सो बाइंड टू द active site of the enzyme, and when they bind to the active site of the enzyme, obviously they are going to block the active site, and as a result the substrate cannot bind to the enzyme, resulting in decrease, no catalysis, there will be no reaction. So that is going to decrease enzyme activity. तो ये जो कॉम्पिटिटिव इनहिबिशन है किस में यूज होता है? इन द कंट्रोल ऑफ बैक्टीरियल पैथोजन. सो वी हैव सर्टेन ड्रग्स व्हिच वी कॉल एज सल्फा ड्रग्स, एंड दोज सल्फा ड्रग्स विल एक्ट एज अ कॉम्पिटिटिव इनहिबिटर फॉर सम एंजाइम्स जो यूज होता है फॉर फॉर्मेशन ऑफ फॉलिक एसिड. किस में? माइक्रोब्स में. अगर फॉलिक एसिड नहीं बनता है माइक्रोब्स में, एज अ रिजल्ट द माइक्रोब्स will be killed and they will be destroyed by these sulfa drugs that we want in case of diseases. Next is non-competitive. So non-competitive inhibition में क्या है? जो inhibitor है उसका कोई structural resemblance नहीं होगा किसके साथ? substrate के साथ. Competitive में हमने देखा था कि दोनों का matching molecular structure है, but यहाँ पे जो inhibitor है it has no resemblance with the substrate. Since it has no resemblance with the substrate, hence ये bind नहीं करेगा किसके साथ? active site of the enzyme के साथ. फिर ये bind कहाँ पे करेगा? एक और site होगा enzyme में, वहाँ पे ये bind करेगा. and चाहे उस enzyme में substrate भी bind हो सकता है और it can be without the substrate. But जब ये inhibitor bind करेगा at some other site, so what it is going to do, it is going to induce change in the enzyme, it is going to induce change in the active site. So as a result, अगर even if the substrate has got attached to the active site, फिर भी जो active site है वो function less efficiently करेगा. So hence the rate of reaction is going to decrease. So what we have seen in non-competitive inhibition, the inhibitor has no structural resemblance and does not bind at the active site of the enzyme. But where does it bind? It is going to bind at a site other than the active site with or without the substrate. And what it is going to do? The inhibitor, it is going to change the conformation of the enzyme as well as the active site. Even if the substrate binds to the active site functions less efficiently, hence the rate of reaction decreases. So इस case में increasing of the substrate concentration will have no effect. So here Km will remain the same, but Vmax is going to decrease. Non-competitive inhibition can be reversible or irreversible. और एक example irreversible का दिया है, like you know cyanide causes death. Why cyanide causes death in an individual? Because it acts as a non-competitive inhibitor. It is going to combine with the copper ions of the cytochrome oxidase, and it's a respiratory enzyme, and that is going to cause inhibition of the cellular respiration, and as a result death occurs. तो ये तो normal reaction हमने दिखाया है कि normally enzyme binds to the substrate at the active site and the products are formed. But inhibition के लिए ये एक और site है, this is the active site, this is some other site जहाँ पे inhibitor bind करेगा. और जब ये inhibitor bind करता है तो ये change करेगा structure को, conformational change लाएगा enzyme में, and as a result of the inhibitor attached to the enzyme तो active site में थोड़ा change होगा. So as a result the inhibitor cannot bind to the enzyme. The inhibitor is going to prevent the binding of the substrate by changing the shape of the active site. या अगर ये bind भी हो गया है, it has got attached here and the inhibitor binds here, still because it is going to cause a change in the active site, so it will function less efficiently. Now coming to allosteric inhibition. सो एलोस्टेरिक इनहिबिशन किस में होता है? एलोस्टेरिक एंजाइम्स जो इन्वॉल्व होंगे मेटाबॉलिक पाथवेज में। अब इन एंजाइम्स में क्या होता है, एक एडिशनल साइट होता है, जैसे हमने प्रीवियसली अभी देखा नॉन कॉम्पिटिटिव में, बट इस साइट को क्या बोलते हैं हम? एलोस्टेरिक साइट in addition to the active site. So allosteric inhibition is reversible, what we say as irreversible non-competitive inhibition. Same mechanism है but reversible है। तो नॉन कॉम्पिटिटिव हमने देखा दोनों हैं, रिवर्सिबल एज वेल एज इरिवर्सिबल। और जो यहाँ इनहिबिटर है, आई सेड जस्ट नाउ दीज आर एंजाइम्स दैट यू फाइंड इन अ मेटाबॉलिक पाथवे। और एक मेटाबॉलिक पाथवे में अगर प्रोडक्ट ज्यादा बनता है, it is more, that is going to act as a inhibitor. So this is one of the mechanisms of controlling enzyme activity. So when that acts as an inhibitor, that is going to put an end to the formation of that product. When that product is used, then this inhibition is lifted. So here the inhibitor, what it is called as modulator. So what it is going to do, it's going to bind with the allosteric enzyme at which site, which we said as the allosteric site, a site other than the active site. So when this inhibitor, which is usually what we said, a product or an intermediate of a metabolic pathway, then it's going to change when it binds to this enzyme at the allosteric site. It is going to cause changes in the enzyme. What we say as allosteric changes is going to change the conformation of the enzyme, and as a result change the conformation of the active site. So that is going to prevent the binding of the substrate molecule to the enzyme. एग्जाम्पल एक है दिया है, फीडबैक और एलोस्टेरिक इनहिबिशन ऑफ द एक्टिविटी ऑफ द एंजाइम hexokinase by glucose 6 phosphate, the product of the reaction it catalyzes. So अभी जैसे नॉन कॉम्पिटिटिव में हमने दिखाया था, ऑलमोस्ट सेम है कि इनहिबिटर which is a product of the metabolic product of a metabolic pathway, तो वो जब बाइंड करता है इसमें, that is going to cause a change in the active site. When it causes a change in the active site, that is going to prevent the substrate from binding to the enzyme. So as a result stop enzyme activity. और जब ये अभी ऊपर जो एग्जाम्पल दिया है ग्लूकोज का, ग्लूकोज 6 फॉस्फेट का, कि ये इस रिएक्शन में ग्लूकोज इन प्रेजेंस ऑफ ATP will be converted into which, what is the product? Glucose 6 phosphate. कौन सा एंजाइम कैटलाइज करता है इस रिएक्शन को? हेक्सोकाइनेस. अब what products will be formed? Glucose 6 phosphate and ADP. That means इसका जो phosphate है किसके साथ अटैच होगा? ग्लूकोज को. at which position? 6th position. अब जब ये excess form होगा, when there is excess of glucose 6 phosphate formed, then this is going to act as an inhibitor. That means this is going to attach where? Here. That means glucose 6 phosphate will get attached to the enzyme. What will be the enzyme? Hexokinase. As a result of attachment of glucose 6 phosphate to the enzyme hexokinase, that is going to inhibit, what are the two initial reactants? Glucose and ATP. That means दो sides होंगे, glucose के लिए ATP के लिए. वो नहीं होगा. That will going to prevent the glucose and ATP from binding to the active sites of the enzyme. तो अगर ये दो चीजें बाइंड नहीं करेंगे to the enzyme hexokinase, as a result enzyme activity will stop. तो जब तक ये प्रोडक्ट यूज नहीं होता है, when the concentration of this product decreases, then this inhibition is lifted and this reaction will go in the forward direction. That means glucose will be converted into glucose 6 phosphate. So this is an example of an allosteric inhibition. Now coming to the last part of this, that is classification and nomenclature of enzymes. So enzymes, you have read so many and all, you must have always observed that end में enzymes में क्या लिखते हैं? ASA, like sucrase, lactase, that shows it is an enzyme. So ASA is added to the name of the substrate. दे मॉडिफाई या कौन सा टाइप ऑफ रिएक्शन ये कैटलाइज करता है. सो इसके बेसिस अब फिर एक यूनियन, इंटरनेशनल यूनियन ऑफ बायोकेमिस्ट्री IUBM, जो फिर लेटर मॉडिफाई हो गई है इनटू इंटरनेशनल यूनियन ऑफ बायोकेमिस्ट्री एंड मॉलिक्यूलर बायोलॉजी IUBMB, they have classified enzymes into 6 major classes. और इन्होंने, दैट इज IUB ने 4 डिजिट का नंबर दिया है, EC नंबर, विच वी से एज एंजाइम कमीशन नंबर, ऑन द बेसिस ऑफ द रिएक्शन इट कैटलाइजेस, जिसमें क्लास होगा, सब क्लास होगा, सब डिविजन होगा सब क्लासेस का, और रिएक्शन इट एंड द रिएक्शन इट कैटलाइजेस. सो एक एग्जाम्पल जैसे सल्फा अल्फा एमाइलेज है, उसमें 4 डिजिट का नंबर है. यू देयर इज नो नीड टू, that means these are 32111 और 3 कौन सा है, will represent the chemical reaction it catalyzes. अभी जो नेक्स्ट हम क्लासिफिकेशन करेंगे ये इसी पे बेसिस है. that means you have to classify the enzymes like is given in this video according to which IUB classification. So you have to write in this particular order only, you cannot change it. What I want to say is class 1. So class 1 हमेशा कौन सा होगा? So पहला डिजिट हमेशा इन एंजाइम्स के लिए क्या होगा? 1. उधर क्या था? 3, because it belong to hydrolases. So class 1 are what? Oxyreductase dehydrogenase. So class 1 हमेशा यही लिखना है. You can't change के randomly अब कोई भी क्लास लिखोगे. No. Class 1 will be IUB के हिसाब से ऑक्सीडोरिडक्टेस और डिहाइड्रोजनेस. तो ये व्हाट डज इट शो? ऑक्सीडोरिडक्टेस. व्हाट आर दीज? रिडॉक्स रिएक्शंस, जहां पे दे आर गोइंग टू कैटलाइज ऑक्सीडेशन एंड रिडक्शन रिएक्शंस वेयर ईदर एन इलेक्ट्रॉन और अ प्रोटॉन इज ट्रांसफर्ड फ्रॉम वन सबस्ट्रेट टू द अदर. तो जनरली हैव एन NADP और NAD एज कोफैक्टर्स. जैसे कि यहाँ AH प्लस B, तो A प्लस BH हो गया, तो दिस हैज गॉट रिड्यूस्ड, या ऑक्सीजन अटैच होगा, सो AO, A प्लस O, दिस हैज गॉट ऑक्सीडाइज. सो इसमें ऑक्सीडेसेस होंगे, दैट मीन्स दैट इज गोइंग टू ट्रांसफर ऑक्सीजन, फॉर्मेशन ऑफ वाटर. रिडक्टेसेस होंगे, डिहाइड्रोजनेस होंगे, दैट इज रिमूवल ऑफ H2. पेरोक्सीडेस एंड कैटलेस, जो कॉज करता है ब्रेकडाउन ऑफ हाइड्रोजन पेरोक्साइड इनटू वाटर एंड ऑक्सीजन. क्लास 2 अब कौन सा आएगा? ट्रांसफरेसेस. सो ट्रांसफरेसेस क्या कैटलाइज करेगा? ट्रांसफर. व्हाट ट्रांसफर? एक फंक्शनल ग्रुप्स का फ्रॉम वन सबस्ट्रेट टू अनदर. सो AB plus C, that means A, this will get transferred to B, so A plus BC. जैसे एग्जाम्पल्स कैसे हैं? ट्रांस अमाइनेस. अमाइनेस व्हाट इज गोइंग टू ट्रांसफर? अमाइनो ग्रुप एक सबस्ट्रेट से दूसरे सबस्ट्रेट पे. ट्रांस एसिटाइलेस क्या ट्रांसफर करेगा? एसिटाइल ग्रुप. Kinases, what's going to transfer? A phosphate group from the ATP. So these are some examples of transferases. Class 3 जो हमने initial बोला था alpha amylase का, what is that? Hydrolases. So that is why first digit क्या था? 3. So class 3 is hydrolases that you have read in your digestive enzymes. What they do? ब्रेकडाउन हो करते हैं सबस्ट्रेट का बाय विद द हेल्प ऑफ वाटर, बाय एडिशन ऑफ वाटर. किस रीजन में? या तो एस्टर बॉन्ड होगा, या ईथर, या पेप्टाइड बॉन्ड, या CC बॉन्ड, C हेलाइड और PN बॉन्ड्स में. सो दे आर गोइंग टू ब्रेक दिस एंड these bonds and result in the breakdown of the substrate molecule. जैसे कि कार्बोहाइड्रेसेस है, ग्लाइकोसाइडेस है जो ब्रेक डाउन करता है लैक्टोज को इनटू ग्लूकोज, गैलेक्टोज. एस्टरेसेस है जिसमें लाइपेसेस आते हैं, फॉस्फेटेस आते हैं. अमाइडेसेस है, amide bond breakdown. Proteases है, peptide bond breakdown. So these are some examples of hydrolases. So that's what we have shown. AB plus water. So this water is going to break these bonds and into its constituents. क्लास 4 कौन सा ग्रुप ऑफ एंजाइम्स है? दैट इज लाइसेस. ये भी ब्रेक डाउन करता है स्पेसिफिक कोवलेंट बॉन्ड्स का लाइसेस, but without using water, without hydrolysis. So but in this process ये क्या produce करता है? Double bonds. New double bonds will be formed or a new ring will be formed. Like we have shown here, this is the compound that is going to be broken down by lysis. So double bond CC attach होगा by double bond and XA will be separated out from this substrate. Example जैसे कि decarboxylases है, decarboxylases है, that is going to remove carbon dioxide from the amino group. So this is one example I've given. Next class is class 5, isomerases, isomers. So a molecular rearrangement, that means converting a molecule of one isomer into another. So what do isomerases do? They are going to catalyze molecular rearrangement of a substrate to form optical, geometrical or positional isomer. जैसे AB होगा तो BA में change होगा तो इसका isomer बनेगा. एग्जाम्पल जैसे कि एलानिन रेसमेस है, वो कैटलाइज क्या कन्वर्जन करता है? कन्वर्ट alanine into its mirror form, isomeric form, that is D-alanine. या mutases है, एक ग्रुप है isomerase क्या, जो शिफ्ट करेगा पोजिशन ऑफ अ ग्रुप, लाइक ग्लूकोज 6 फॉस्फेट को किस में कन्वर्ट करेगा? टू ग्लूकोज 1 फॉस्फेट. दैट मीन्स फॉस्फेट को कहाँ से शिफ्ट किया? 6 पोजिशन से फर्स्ट पोजिशन ऑफ द ग्लूकोज मॉलिक्यूल. सो दिस आर ऑल्सो अनदर एग्जाम्पल ऑफ आइसोमरेज एंड देयर आर मेनी मोर. Last class, class 6, ligases or synthetases. Ligase जोड़ना, synthesis जोड़ना. So what are these enzymes going to do? They are going to catalyze the joining of two substrate. And join के लिए क्या चाहिए? By utilizing, should be Y here, utilizing energy from ATP. So like they are going to join bonds किसके? CO के बीच में join करेंगे, CS के बीच में CN, PO bonds, etc. Obviously by removal of water. And what are these? Condensation reactions. एक example जो frequently you are going to read later on also, that is DNA ligase. What it is going to join? Two fragments of DNA by formation of phosphodiester bond. So this was all about the various classes of the enzyme, and you have to write these classes in this particular order, because this is the order that IUB has given. So that's all for today, and hope you understood what is being said in this video. In case you have any doubt, you should come up and contact us. Thank you and God bless you all.